Glucagon, this is alpha, this is insulin is a beta, somatostatin is a delta, right? The glucagon increases secretion of the insulin, and this somatostatin inhibit the secretion of insulin, and this also inhibit the secretion of glucagon. Okay, it's a cyclic reaction. Mechanism of action of insulin. Insulin act on tyrosine kinase receptor. This, which is a heterotatamic glycoprotein consisting of two extracellular alpha and these two transmembrane beta subunits linked with the disulfide bonds binding with the next is binding with the insulin on the alpha subunit induce the internucleation of the receptor along with the bond insulin. This thus activate the tyrosine activity of beta unit. After it, the beta cells get into the autophosphorylation, which that furtherly they activate the insulin receptor subprotein through the phosphorylation, which results in the activation of all the secondary messenger pathway like MAPK and PI3K, and this that state which is result in the synthesis of GLUT4 transporter, which is translocate on that cell membrane for the another glucose transport. Okay, when this insulin bind into the alpha subunit, it internucleates. Okay, after internucleation, it first of all it has the alpha subunits and beta subunits. Okay, after internucleation, that is come into the cell. Okay, that internucleation occur. It's a poly cell. Okay, autophosphorylation takes place. This is a trimeric in nature. Tyrosine kinase increase the activation of this substrate called insulin receptor substrate. After increasing this, that which cell present on this is two pathways, MAPK and IP3K pathway. This MAPK causes the proliferation of protein, means cell difference. Next, here PI3K pathway, this synthesis the gene expression, increase the gene expression, protein synthesis, GLUT4 transport. For this is a protein synthesis occur, GLUT4 transporter increases, so that's again inserted into the membrane for the more glucose into the cell. So this is a mechanism of action of an insulin. Okay, this is a normal mechanism that occurs. Pharmacological action. The insulin is a profound action on the metabolism of carbohydrate, fat, protein, and facilitate the entry of glucose into the cells of the body. Glucose entry into the RBC, WBC brain cell cause occur in the interpreted insulin. Insulin is an inhibition hepatic glycolysis and glucogenesis lipolysis in the adipose tissue. Insulin enhances the entry of the amino acids into muscle cells, thereby promote the synthesis of the protein in muscle. Insulin facilitates the glycogen synthesis in the liver, muscle